Hello, I'm Luca Motoneuron from the University of New South Wales in Sydney, Australia, and in this talk I will present the latest work that we've done in neuromusculoskeletal modeling with colleagues from Imperial College London, where I was previously based, and in particular I will present some of the work being developed by Arnault Caillet during his PhD. In this presentation, I will try to highlight the novelty of our new modeling technique with respect to the main components of a neuromechanical investigation. In particular, I will focus on the mechanical representation of the musculoskeletal system and on the neural control that, given a certain task, allow us to investigate how real-world behavior is produced. In our modeling work, the anatomy and the mechanical function of the musculoskeletal system are represented by a musculoskeletal model, usable in fully dynamic simulation of motion. The neural controls considered in this presentation, on the other hand, are based on experimental electromyographical recordings. It is important to describe the state-of-the-art in neural controls for fully understanding the following of the presentation. Classically, standard bipolar surface EMG signals have been processed to obtain a neural excitation E that was then processed through an excitation activation dynamics. The final output of these transformations is an activation used as an input for the contractile element of a lumped parameter hill type model, often representing an entire skeletal muscle. In 2017, Motoneuron specific spike trains obtained from the composed high-density surface EMG signals were used by Sartori and co-workers to derive the muscle active state. However, these multiple spike trains were, like in previous studies, merged into a single neural control and fed to a single hill-type model, again representative of an entire skeletal muscle. We believe that the composed high-density surface EMG signals provide enough information to control individual motor units. More in detail, we are convinced that when a certain number of spike trains for individual motor neurons are identified from high-density surface EMG, they can be used as controls for the individual motor units that they innervate, providing their individual active states. In other terms, we believe that a muscle model with a resolution at the motor unit level could be controlled, rather than a single hill-type muscle model. The aim of this presentation is therefore introducing a novel motoneuron-driven muscle model with a motor unit resolution in muscle force prediction. I will also present a use of this model in a personalized neuromusculoskeletal model simulation where the muscle architecture is represented using an advanced volumetric approach that we developed in 2020. And you can see an example on the right. The combination of these neural and anatomical models is currently the highest level of personalization achievable in vivo with non-invasive techniques. We want to demonstrate the potential of this modeling approach for neuromechanical research, meaning understanding how the spinal motor neuron activity influences human motor function. But the relevance of this research potentially extends also to the brain-machine interfaces and prosthetic design. The first aspect that we considered was the mechanical representation of the musculoskeletal system. For our investigation, rather than employing a generic model based on cadaveric data, as normally done in neuromechanical studies, and then linearly scale it based on anthropometry, we collected medical images for the participant of our study. Subsequently, bone and muscle tendon geometries were segmented from the images to obtain a three-dimensional representation of these anatomical structures. Our specific interest here is on the tibialis anteriors, which is the skeletal muscle that we will focus on. The musculoskeletal model was built using automatic tools. The skeletal kinematic model was created in seconds using the staple toolbox developed by Jean-Baptiste Renaud from the Marseille University and myself last year, which was then extended to non-rigidly map on the tibia its typical muscle attachment areas. These areas were obtained originally from a previously published cadaveric dataset. The anatomical model of the tibialis anterior was obtained using another automatic technique, developed in collaboration with Joseph Cote of the University of West Bohemia. The technique consists in mapping a template of the muscle fiber arrangement on the muscle segmented geometry, obtaining a highly discretized muscle tendon representation. Each geometrical path will be considered as a motor unit actuator in our final model. The final musculoskeletal model included a tibialis anterior with 400 muscle fibers, number that will be matched by the neural controls. The muscle can produce a maximum isometric force estimated from the segmented muscle volume. The muscle fibers present a common tendon and the moment arm estimated with respect to a personalized ankle axis. The experimental protocol consists of the participant performing isometric trapezoidal contractions while the activity of the tibialis anterior is recorded using high-density surface EMG and the ankle joint torque is measured, normalized with respect to that obtained during maximum voluntary contraction. The ankle is positioned at 10 degrees of front flexion. In first instance, we assumed that the tibialis anterior was generating the totality of this torque. The electromyographical recordings were then decomposed in individual motoneuron spike trains by blind source separation. With careful control of the experimental setting and optimization of the setup, we were able to obtain as many as 82 motoneurons in certain trials, but also used as input of our model 32 motoneurons, and in other trials 52 and 21. We will use these different outcomes to test the robustness of our neuromechanical model with respect to its inputs. The first step of our model consists in a reconstruction of the activity of the entire motoneuron pool innervating the muscle of interest. This is performed through a method developed by Arnault Caillet, who will explain it in details during this presentation in this same session. Using the force trace obtained transforming the recorded ankle joint torque through the known muscle moment arm, we were able to identify the force recruitment threshold of each motoneuron, so ranking them. Using their spike trains, on the other hand, we could compute the common synaptic current and use it as input to computational motoneuron models that were calibrated on the experimental discharge rates, exploiting empirical relationships formulated by Arnault after analyzing a large amount of literature data. As a result of this approach, we can estimate the motoneuron size distribution across the entire motoneuron pool and use it to create 400 virtual motoneurons with realistic firing behavior. The number 400 was estimated based on information available in the literature. After reconstructing the firing behavior of 400 virtual motoneurons, we also developed a detailed excitation and activation dynamics to process each of their spike trains, combining elements from Atz's muscle model from 1977 and 1978 with original contributions implemented again by Arnault. First, we modeled the propagation of the action potentials both on the nerve and on the muscle, followed by the release of free calcium in the muscle fibers and the binding with troponin, which frees the myosin attachment sites. Finally, we computed an active state for each motor unit that we can use as controls for the 400 hill-type models. It is worth noting that these are interconnected models where the output of one step is the input of the following one. The motor unit active states for the 400 considered motor units were scaled during the contraction dynamics using an activation-dependent normalized force length relationship derived from Lloyd-Bézier. The normalized lengths were computed from the corresponding fibers in the musculoskeletal model, of which you can see the length distributions here. The distribution of the motor unit tetanic forces was also estimated from the subject-specific maximum isometric force, using a relationship derived from the literature. The motor unit forces were finally summed to predict the whole muscle force, which we could now compare to the experimental force trace. This is an example of simulation using motoneuron spike trains as input. The model is simulating a ramp contraction of increasing load, and you can see how the individual motor units, each represented by a single line of action, are recruited while the ankle torque increases, until the majority of the muscle volume is contributing to the produced force. As I already hinted, our simulations were validated by comparing the simulated muscle force against the force trace derived from the experiments. The agreement was quantified in terms of coefficient of determination and normalized root mean square error, considering 4 cases with 21, 32, 52, and 82 motor units respectively, as mentioned when describing the high-density surface EMG decomposition results. According to these metrics, The muscle force obtained simulating the complete motoneuron pool with 400 virtual motoneurons in red matched very well the experimental force trace. We also found that the muscle force predictions were more accurate after the entire motoneuron pool was reconstructed than using just the experimental identified motoneurons, for which the results are shown in blue. Finally, regardless of how the motoneuron pool was represented in the models, we found better validation metrics when a larger number of motoneurons were identified experimentally, with normalized root mean square error reducing almost 3 times from 82 to 21 motoneurons. It is also worth noting that these results were obtained without any musculoskeletal parameter calibration, which is typical of previous EMG-based approaches. Our model can currently simulate only isometric muscle contractions due to the technical challenging in high-density surface EMG decomposition. The accuracy of the predicted common control, as you have also seen in the results, also depends on the number and spread of the identifying motoneurons obtained from the decomposition itself. In terms of future development, we are currently working towards the application of this modeling approach to muscles other than the tibialis anterior, also when key information like the distribution of motor unit recruitment force thresholds and motor unit tonic forces have never been measured. I thank you for your attention, and I will be more than happy to answer any question you might have. Thank you very much.